Plant growth is a permanent and irreversible increase in a plant's size, caused mainly by cell division and cell enlargement. Why is it called open-ended? Because plants have meristems that keep dividing throughout life, allowing continuous formation of new roots, shoots and leaves. This is called indeterminate growth. Types of growth in plants. Plants show two main types of growth: indeterminate growth and determinate growth. Let's understand them one by one. First is indeterminate growth. Indeterminate growth means that the plant continues to grow throughout its life. This is possible because plants have special tissues called meristematic tissues. These tissues contain actively dividing cells that can produce new cells continuously. For example, the root and shoot systems keep growing because their tips contain meristematic tissues. So, remember, indeterminate growth means growth continues, Now, let's understand determinate growth. Determinate growth means that growth stops after reaching a specific size or stage. For example, leaves and flowers generally stop growing once they reach their mature size. So, remember, determinate growth means growth stops after maturity. Easy way to remember. Think of a plant as a whole. A plant generally shows indeterminate growth because its roots and shoots can keep producing new growth. But an individual organ such as a leaf or flower, may show determinate growth because it stops growing after reaching maturity. So the key point is, plant right pointing arrow generally indeterminate growth. Individual organ right pointing arrow may show determinate growth. In one line. Indeterminate means growth continues, while determinate means growth stops after a particular stage. Regions of plant growth. Easy explanation: plants do not grow equally from every part. Most plant growth occurs in special tissues called meristems. Meristematic cells have the ability to divide continuously and produce new cells. There are three important types of meristems: apical meristem, intercalary meristem, and lateral meristem. First, let's understand the apical meristem. Apical meristems are present at the tips of roots and shoots. Their main function is to increase the length of the plant. For example, when a root grows deeper into the soil or a shoot grows taller, this growth is mainly due to the activity of apical meristems. This increase in the length of the plant is called primary growth. Next is the intercalary meristem. Intercalary meristems are generally found at the bases of internodes or at the bases of leaves especially in grasses. They help in increasing the length of internodes. This is why grasses can continue growing even after their tips are cut. Finally, we have the lateral meristem. Lateral meristems are present along the sides of stems and roots. Their main function is to increase the girth or thickness of the plant. This type of increase in thickness is called secondary growth. Two important lateral meristems involved in secondary growth are vascular cambium and cork cambium. The vascular cambium produces secondary xylem and secondary phloem, helping the stem and root become thicker. The cork cambium produces cork cells and contributes to the formation of the protective outer covering. So, remember this simple concept. Apical meristem increases length. Intercalary meristem increases length, especially at internodes. Lateral meristem increases girth or thickness. And the easiest way to remember the two types of growth is primary growth means growth in length. Secondary growth means growth in thickness or girth. In short, tips help the plant grow longer, while sides help the plant grow thicker. Plant growth takes place in three main phases. The first phase is the meristematic phase. In this phase, the cells are actively dividing and producing new cells. These cells have thin cell walls, dense cytoplasm, and a prominent nucleus. They have very small vacuoles, or sometimes vacuoles may be absent. The metabolic activity of these cells is also very high because they are continuously dividing. The second phase is the elongation phase. After cell division, the cells start increasing in size. During this phase, the cells become longer and larger. Large vacuoles are formed inside the cells. The cell wall also undergoes deposition and modification. As a result, the length of the plant part increases. The third phase is the maturation phase. In this phase, the cells reach their maximum size. The cells become specialized and start performing specific functions. This process is called differentiation. The cells also undergo structural and functional changes according to the role they have to perform. So the complete sequence of plant growth can be remembered as meristematic phase followed by elongation phase followed by maturation phase. In simple words, first the cells divide, then the cells increase in size, and finally the cells become specialized. This is how plant growth takes place from newly formed cells to fully mature and specialized cells. Plant growth can be measured in different ways depending on which part of the plant is growing. We can measure growth by observing the increase in length of a plant part such as the root or stem. We can also measure the increase in area, for example the increase in leaf area. Growth can be measured by an increase in volume when the size of a plant organ becomes larger. Another method is measuring fresh weight, which tells us the weight of the plant including the water present in its tissues. Dry weight can also be measured by removing the water from the plant tissue and then measuring its remaining mass. Growth can also be measured by counting the increase in the number of cells. For example, if we want to measure the growth of a root, we can measure its length at regular intervals of time. Suppose a root is 5 centimeters long at the beginning and after 5 days it becomes 10 centimeters long. This increase in length shows that the root has grown by 5 centimeters during those 5 days. Therefore, growth means a measurable increase in size, mass, volume, area, or cell number over a period of time. Arithmetic growth is a type of plant growth in which only one daughter cell continues to divide while the other daughter cell stops dividing and starts to mature. Because only one cell keeps dividing, the growth takes place at a constant rate. This means that the plant part increases in length by almost the same amount during each equal period of time. For example, if a root grows 2 centimeters every day, then after 1 day it grows 2 centimeters, after 2 days it grows 4 centimeters, and after 3 days it grows 6 centimeters. The formula for arithmetic growth is LT equals L0 plus RT. Here LT represents the length of the plant part at a particular time. L0 represents the initial length of the plant part, R represents the constant growth rate, and T represents the time period. Since the growth rate remains constant, the graph of arithmetic growth is a straight line. So the main point to remember is that in arithmetic growth only one daughter cell continues to divide and the growth occurs at a constant rate, producing a linear or straight line growth curve. Geometric growth means that both daughter cells produced after cell division continue to divide further. In the beginning the growth is slow because the number of actively dividing cells is still low. As cell division continues, the number of cells increases rapidly and the growth becomes much faster. This type of growth produces an exponential growth curve. Geometric growth can be understood in 3 main phases. The first phase is called the lag phase. During this phase growth is slow because cells are preparing for active division and enlargement. The second phase is called the log phase or exponential phase. During this phase cells divide very rapidly and cell size also increases. As a result, the overall growth becomes very fast. The third phase is called the stationary phase. During this phase the growth rate gradually decreases and finally becomes almost constant. The mathematical expression for geometric growth is W1 equals W0 into e raised to RT. Here W1 represents the final growth, W0 represents the initial growth, R represents the growth rate, and T represents the time period. So in simple words, geometric growth means slow growth in the beginning followed by very rapid growth and finally a stage where growth becomes almost constant. Absolute growth rate means how much a plant grows in a given amount of time. In simple words, it tells us the total increase in the size of a plant per unit time. For example, suppose a plant is initially 10 centimeters tall. After 5 days its height becomes 20 centimeters. So the total growth is 20 minus 10, which is equal to 10 centimeters. Now we divide this total growth by the time taken. So 10 centimeters divided by 5 days gives 2 centimeters per day. Therefore the absolute growth rate of the plant is 2 centimeters per day. This means that on average the plant increased its height by 2 centimeters every day. Relative growth rate means how much a plant grows compared with its original size in a given amount of time. For example, suppose one plant is 10 centimeters tall and another plant is 100 centimeters tall. If both plants grow by 10 centimeters in 1 week, their absolute growth is the same because both have increased by 10 centimeters. But their relative growth is different. The first plant has doubled its size from 10 centimeters to 20 centimeters, so its growth is very large compared with its original size. The second plant has increased only from 100 centimeters to 110 centimeters, so its growth is much smaller compared with its original size. Therefore, relative growth rate takes the initial size of the plant into account. This is why relative growth rate is useful when we want to compare the growth of plants or plant parts that have different starting sizes. In simple words, absolute growth tells us how much the plant has grown, while relative growth tells us how much the plant has grown in comparison to its original size. Conditions necessary for plant growth. For healthy growth and development, plants need several suitable conditions. These conditions can be divided into two main categories. The first category is external factors. The second category is internal factors. Let us first understand the external factors. The first important factor is water. Water is essential for plant growth because it helps in cell expansion and maintains the turgidity of plant cells. Water is also required for photosynthesis, which is the process through which plants prepare their food. It also helps in the transportation of minerals and other substances throughout the plant. The second important factor is oxygen. Plants need oxygen mainly for respiration. During respiration, plants release energy from food, and this energy is required for different activities such as growth, cell division, and other metabolic processes. The third important factor is nutrients and minerals. Plants require different mineral nutrients for proper growth and development. These nutrients are used to form important substances such as proteins, nucleic acids, enzymes, cell walls, and cell membranes. Without proper mineral nutrition, plants may show poor growth and deficiency symptoms. The fourth important factor is temperature. Every plant requires a suitable temperature range for normal growth. There are three important temperature points. The minimum temperature is the lowest temperature at which growth can occur. The optimum temperature is the temperature at which growth occurs most efficiently. The maximum temperature is the highest temperature at which growth can occur. Temperatures below the minimum or above the maximum can slow down or stop plant growth. Light is another important external factor. Light is essential for photosynthesis and also affects several processes related to plant growth and development. Now let us understand the internal factors. The first internal factor is genetic factors. The genetic makeup of a plant controls many characteristics, including its growth pattern, size, development, and response to environmental conditions. The second internal factor is plant growth regulators. Plant growth regulators are chemical substances produced naturally by plants that control and coordinate different growth and developmental processes. Examples include auxins, gibberellins, cytokinins, ethylene, and abscisic acid. Therefore, healthy plant growth depends on a proper balance between external environmental conditions and internal factors. Differentiation is the process by which cells produced by meristematic tissues change their structure and function and become specialized to perform a specific role in the plant. For example, a meristematic cell can develop into a xylem cell. During this process, the cell undergoes several changes in its size, shape, structure, and internal organization. A xylem cell develops specialized features that help it transport water and minerals from the roots to other parts of the plant. In simple words, differentiation means that a general cell changes into a specialized cell with a specific function. For example, a meristematic cell can become a xylem cell for water transport, a phloem cell for food transport, or a specialized cell of the epidermis for protection. Dedifferentiation is a process in plants in which living permanent cells regain their ability to divide. Normally, permanent cells become specialized and perform specific functions, and they usually lose their ability to divide. But under certain conditions, these specialized cells can become capable of cell division again. This process is called dedifferentiation. For example, some permanent cells can develop into interfascicular cambium or cork cambium. These tissues regain the ability to divide and produce new cells. So we can remember dedifferentiation in a very simple way. A specialized cell regains its ability to divide. In short, dedifferentiation means specialized cells become capable of cell division again. Redifferentiation is the process in which cells that have regained the ability to divide through dedifferentiation again become specialized and lose their ability to divide. The process can be understood in simple steps. First, a meristematic cell undergoes differentiation and becomes a permanent specialized cell. Then, under suitable conditions, this permanent cell undergoes dedifferentiation and regains the ability to divide. After that, the newly formed cells undergo redifferentiation. During redifferentiation, these cells become specialized again and perform a specific function. So the sequence is: meristematic cell, differentiation, permanent specialized cell, dedifferentiation, cell regains the ability to divide, redifferentiation, new specialized cell. In simple words, redifferentiation means a cell that has regained its ability to divide becomes specialized again. Development means all the changes that happen in a plant or organism throughout its life cycle. It includes growth, differentiation, and other developmental processes. Growth means an increase in size or mass. Differentiation means cells become specialized to perform specific functions. Development is therefore a broader process that includes both growth and differentiation. In simple words, development means the complete process through which an organism grows, changes, and becomes mature. Developmental plasticity means the ability of a plant to change its growth and development according to the environmental conditions around it. In simple words, a plant can change its structure and form when the environment changes. One important example of developmental plasticity is heterophyly. Heterophyly means the formation of different types of leaves on the same plant. For example, the leaves produced during the juvenile stage of a plant may have a different shape from the leaves produced during the adult stage. This difference in leaf shape is called heterophyly. Therefore, developmental plasticity helps plants adjust their growth and development according to different environmental conditions. Plant growth regulators are small organic substances that control and influence the growth and development of plants. They help plants perform different activities such as cell division, cell enlargement, flowering, fruit development, fruit ripening, seed germination, and seed dormancy. Plant growth regulators can be naturally produced by plants or they can be made artificially and are called synthetic plant growth regulators. There are 5 major groups of plant growth regulators. The first group is auxins. Auxins mainly promote cell elongation and help in the growth of shoots and roots. They are also responsible for apical dominance, which means the main shoot suppresses the growth of side branches. Auxins can also help in root formation, fruit development, and formation of seedless fruits. The second group is gibberellins. Gibberellins mainly promote stem growth and increase the length of plant internodes. They are responsible for a process called bolting in which the stem of certain plants grows rapidly before flowering. Gibberellins also help in seed germination and can help break seed dormancy. The third group is cytokinins. Cytokinins mainly promote cell division in plants. They also help in the development of shoots and delay the aging of leaves. The fourth group is ethylene. Ethylene is a gaseous plant growth regulator. It is mainly responsible for fruit ripening. It also promotes aging of leaves and flowers and helps in the shedding of leaves, flowers, and fruits. The fifth group is abscisic acid or ABA. ABA is mainly known as a stress hormone. It helps plants respond to conditions such as drought. ABA causes stomatal closure, which reduces water loss from the leaves. It also promotes seed dormancy and helps plants survive unfavorable conditions. So we can remember the 5 major plant growth regulators as auxins, gibberellins, cytokinins, ethylene, and abscisic acid. Auxins help in elongation. Gibberellins help in growth and bolting. Cytokinins help in cell division. Ethylene helps in fruit ripening. And ABA helps in stress response and dormancy. Auxin is a plant growth hormone. The word auxin means to grow. The main natural auxin is IAA or indole-3-acetic acid. Other important auxins are EBA and 2,4-D. Auxin promotes cell elongation, especially in shoots. It maintains apical dominance, where the apical bud suppresses the growth of lateral buds. When the shoot tip is removed, lateral branches grow faster. Auxin helps in the formation of adventitious roots. It controls phototropism and geotropism. It promotes fruit development and can produce seedless fruits by parthenocarpy. It also delays the premature dropping of leaves and fruits. In agriculture, 2,4-D is used as a selective herbicide to control broad-leaved weeds. Gibberellins. Gibberellins are plant hormones that mainly promote growth. They were discovered while studying Bakani disease in rice. The most important gibberellin is GA3 or gibberellic acid. It increases stem length, causes bolting, helps break seed dormancy, stimulates alpha amylase enzyme during seed germination, promotes fruit growth, and supports flowering in some plants. Memory trick. GA means grow above. Cytokinins. Cytokinins are plant hormones that promote cell division. The main natural cytokinin is zeatin. They help in cell division, cell differentiation, shoot formation, delay aging of leaves, and move nutrients to actively growing parts. Memory trick. Cytokinin means cell division. Ethylene. Ethylene is the only gaseous plant hormone. Its chemical formula is C2H4. It helps in fruit ripening, promotes aging of leaves and flowers, causes leaf, flower, and fruit drop, influences seedling growth, promotes flowering in pineapple, and increases female flowers in some plants. Memory trick. Ethylene means ripen, age, and drop. Abscisic acid or ABA. Abscisic acid is called the stress hormone. It helps plants survive during drought by closing stomata to reduce water loss. It promotes seed dormancy, bud dormancy, slows plant growth, and helps plants respond to stress conditions like drought and water shortage. Memory trick. ABA means anti-growth, stress, and dormancy. Plant growth regulators control plant growth and development. Growth promoters are auxins, gibberellins, and cytokinins. Auxins help in cell elongation and root formation. Gibberellins increase stem length and help seeds germinate. Cytokinins promote cell division and delay aging. Growth inhibitors are ABA and ethylene. ABA slows plant growth, closes stomata during drought, and causes seed dormancy. Ethylene helps in fruit ripening and also promotes aging and leaf fall depending on the situation. Plant hormones work together and their effects depend on their balance inside the plant. Photoperiodism. Photoperiodism is the response of plants to the length of day and night, especially for flowering. Short day plants flower when the night is longer than a critical period. Examples are chrysanthemum and xanthium. Long day plants flower when the day is longer and the night is shorter. Examples are wheat, spinach, and radish. Day-neutral plants flower regardless of day length. Examples are tomato and cucumber. The uninterrupted dark period is more important than the length of the day. Phytochrome. Phytochrome is a light-sensitive pigment that helps plants detect light. It has two forms, PR and PFR. PR absorbs red light. PFR absorbs far red light. Phytochrome helps control flowering and other responses to light. Vernalization. Vernalization is the process in which low temperature promotes flowering. It is important in plants like winter wheat, winter rye, and some biennial plants. It helps plants flower at the correct season and reduces the vegetative period. Remember, photoperiodism depends on day and night length, while vernalization depends on low temperature. Seed dormancy. Seed dormancy is a resting stage in which a healthy seed does not germinate even in favorable conditions. It can happen because of a hard seed coat, growth inhibitors, an immature embryo, or special environmental requirements. Dormancy prevents seeds from germinating during unfavorable seasons. ABA promotes seed dormancy. Gibberellins break dormancy and promote seed germination. Remember, ABA means dormancy and GA means germination.